as a scientist, decided that one way to handle my own deep and profound anguish over losing my home and perhaps my city was to turn it into an experiment.
There's a significant study from Finland that showed that kids exposed to the musty odor have 2 times the risk of developing asthma later in life. The exposure to the musty smell early in life was the second leading predictor of asthma behind maternal smoking.
The more I have read about the human side of mold exposure, the more I realize that we have huge amounts to learn, because there are psychological aspects and there are neurological aspects that are related to the psychological aspects.
Our next guest today is someone I've been fortunate to call a friend for almost 20 years, Dr. Joan Bennett from Rutgers University. Joan is a true pioneer in the field of mycology and fungal biology. Her groundbreaking research has explored the volatile organic compounds produced by molds, which she has termed volatoxins. These gaseous compounds can have significant impacts on human health, even at extremely low levels. Joan's work is helping shed light on an often overlooked aspect of mold exposure beyond just spores and mycotoxins. Joan's distinguished career has earned her election to the National Academy of Sciences and the American Academy of Arts and Sciences. She's the past editor-in-chief of the esteemed journal Mycologia, past vice president of the British Mycological Society and the International Union of Microbiological Societies, as well as the past president of the American Society for Microbiology and the Society for Industrial Microbiology and Biotechnology. Needless to say, we have a bona fide mold expert here. I'm thrilled to have her join us today at the Got Mold Virtual Summit to help clear the air and debunk a few myths. Please join me in welcoming the brilliant Dr. Joan Bennett.
Thank you for the lovely introduction.
Hey there, sorry to jump in here, but really important. At the end of this video, there's a link that gives you access to a bonus package we put together that has all sorts of great resources. Pro-level notes, audio downloads, a fast start guide, as well as some additional resources from our speakers. You will not want to miss out on that. You can also find the link in the description below or at gotmoldsummit.com. So go claim yours today. Now back to the interview.
You know, Joan, I mean, it's nothing but the facts, right? I mean, you've got quite a storied career. And like I said earlier, we've known each other for a very long time. And I thought it might be fun just to sort of share that story with our audience, because when I first started working with Oreo, our first mold detection dog, we helped instruct a course on mold and dampness in buildings over at Rutgers. And I believe you caught wind of it. And gave my office a call and said, "Hey, I'd really like to meet your dog. And it's okay if Jason comes too."
Basically, right. But I gathered my lab together and we were all set up in a lounge and we were hot to meet the dog. And I remember I brought treats and you wouldn't let me give Oreo any treats because you said Oreo only gets treats if it finds mold.
That's right. That's
right. We didn't have mold in the lounge or the lab that was right to be detecting. It was all hidden in the incubators.
That's right. No, but so she was always she was quite a calling card. And clearly, you know, she brought us together, which is something that I'm eternally grateful for. You know, so in the process of us meeting, you began to show me your work, and I had no idea that it would lead me down a path of curiosity that continues to increase with every passing day. It's an area of research that has not gotten enough attention, but it's one that you've truly pioneered. So, you know, in terms of the microbial volatiles, and just a quick definition on that. So the volatile organic compounds, VOCs, produced by mold, essentially what we're talking about is the musty smell, right?
Yeah. You want to do something really broad with it. The idea of a volatile is the substance that usually, if it's enclosed at room temperature, is a liquid. You think of perfume in a bottle. But if it gets up to the air, it will go into gas phase. And that's why it's not a permanent gas. You know, it's something that's right at the edge. And so when it's given a chance, it goes into the air. And everything we smell is a volatile. if you smell an odor, it is one of these molecules, one of these compounds that has managed to get into the gas phase. And olfaction is really not as well understood even to this day as some other aspects of biology. And so I'm interested in the volatiles made by fungi, but believe me, there are a lot of other volatiles out there. When we smell car exhaust, or when we smell cooking, it smells good, or when we smell air pollution from a nasty factory, and the list goes on and on. When we smell rotting food, when we smell something delicious in the frying pan. Those small molecules that we can detect, those are volatile.
Yeah, very, very powerful. And that's actually what mold dogs are trained to detect when it comes to finding hidden mold. And anyone who's been through a hidden mold issue knows that mold can be kind of tricky to find. And so dogs are trained to detect the source of that odor, because when mold is growing, it's digesting what it's growing on and releasing these essentially digestive gases, right?
Yeah, that's one way of looking at it. They're breaking down compounds, and when the molecules get small enough, they can be released into the air and create these odors. Yes.
So you've coined the term volatoxins, which I'll let you unpack here. So you've had such a pioneering career in mycology. And can you tell us a little bit about what first sparked your interest in studying fungi and volatiles, mold volatiles?
Well, I wish I knew why I was interested in studying fungi. That goes way back. And, there's something about the life form that's so strange. And I was attracted from a very early age, and went to graduate school and actually became a fungal geneticist. I love genetics and I love fungi. My first job out of graduate school was for the Department of Agriculture. And the USDA, U.S. Department of Agriculture, had a major project going on one of the first of the major mycotoxins discovered, fungal toxin, something called aflatoxin. And I spent over 25 years studying that toxin and doing genetics and learning a lot about fungal toxins. And I learned that they are a huge problem in the global food supply, and that there are numerous toxins made by different fungi that can get into things we eat. And the USDA and the Food and Drug Administration work very hard to keep them out of the food supply. So with that background, I was an expert on mycotoxins when somewhere in the 1990s, 30 years ago, people started to get worried about toxins in houses. And there was a lot of publicity about some people who felt they'd been sickened by mold toxins in indoor air. And it's often called thick building syndrome, although there are other names. And I knew enough about mold toxins that I believed that it was really hard to get exposed to enough toxin through the air to get sick. And more than that, the toxins that we knew most about were not made on building materials. They were made on food, cornflakes, peanuts, pistachios, all kinds of food products have mold toxins in them. But when mold is growing on sheetrock or wet books or behind some wall on insulation, it's a different food source. They're different molds, and there are a few toxigenic molds in there, but it's really hard to get exposed to them. And so I'm not necessarily proud of this, but during the 1990s, I accepted a number of consulting cases for insurance companies where I would say, hey, I'm a mold expert, and I don't think this person who claims they're sick in a building has been made sick by fungal toxins. I don't know how long you want me to go this monologue, and
oh yeah, please, yeah.
When I changed, but here I was, a mold expert, a mycotoxin expert, and then I experienced my own mold disaster. I was on the faculty of Tulane University, which is located in New Orleans, Louisiana. And in 2005, New Orleans was struck by a hurricane that caused the levees to fail. And the city, which is largely under sea level, flooded. There was a big lake that flooded in, and my home was one of the ones that flooded. And I didn't get home for a while, but by the time I got home, I call it mold anarchy. There was fungus growing on the entire ground floor of my house, on the walls, on the furniture, on the books, on the rugs. And I had, as a scientist, decided that one way to handle my own deep and profound anguish over losing my home and perhaps my city was to turn it into an experiment. So the first time I went home, I traveled with Petri dishes. I had an N95 mask. I was wearing gloves, and I sampled my house. I was going to try to figure out what fungi were in there. And this is where I had my conversion experience as I was sampling my house, wearing an N95 mask, so I couldn't be breathing any mold spores. I started to feel sick. And it's very hard in words to describe odors. It's something that many of us, I think, have faced when we're trying to talk about smells. So when I try to describe that odor, I'm at a loss for words. It was horrible. It was overpowering. It was foul. It was this terrible stink. And as many odors, as I was in the house sampling, I smelled the terrible odor less and less, but I started to get headaches. I felt nauseated. I felt like every cell in my body was saying, get out of there. So actually what I ended up doing was I put a chair outside, and I'd go and sit outside for a while and take my mask off. And then I'd go back in and I'd sample for another 10 minutes. And it was then that I decided that I'm going to stop studying mycotoxins, and I'm going to start studying these volatile compounds, these smell compounds, these things that I'm able to breathe through a mask, that they're so small that they're not filtered out by the mask. And I met you early on in those studies. Here I am, maybe it took me about a year to set up my lab, maybe 17 years into it. I've only become more convinced since then that some volatile compounds can be toxic. An unexpected finding along the way is that I've also found that some of these volatiles have other activities. And the first time we discovered a fungus that was making a plant model grow better, I almost couldn't believe it, because we've been killing the plants with certain volatiles, and then we grew a certain fungus next to it, and the plant was growing better. And we've since learned that there are volatiles that do some good things out there too. But my original focus, and to this day it continues to be the focus, is that I think that fungi and other microbes and maybe other organisms can make gas phase compounds that make us sick, and hence the term volatoxin.
Thank you very much for that. So I interviewed Dr. Birgitte Andersen from Aalborg University in Copenhagen yesterday, and she does fascinating work with building materials and fungi. And so she describes these compounds, both mycotoxins and volatiles, as chemical messengers for anyone who's willing to listen, essentially. Of course, mostly microbes. And it seems to me that there might be some conversations going on with the plant world as well. And it's funny you mentioned that every cell in your body was telling you to leave. And I've often had the notion that mold makes it uncomfortable in our buildings to make us leave so that it can finish eating the rest of your house, to anthropomorphize it a little bit,
Yeah, well, we have evolved to be sensitive to danger signals, and we accept it in food that, there's certain foods that taste bitter, and hey, I'm not going to swallow that. Well, there are certain smells that tell us, hey, get out of there. And they add a certain, smelly compound to gas leaks where we can't necessarily smell with some of our fuel gases, but it's a very human response to danger, and somehow built into our evolutionary machinery. And for for the problems around indoor mold, though, one of the things that I didn't know much about olfaction when I started this, but I've learned is that we get desensitized. So if you're with this bad odor that tells you to leave for a while, you get used to it and you can overcome it. And at some point you don't even smell it anymore. And I think we've all had that experience with bad smells, that we notice it when we walk into the room and then half an hour later we hardly notice it at all. But that doesn't mean that the odor's gone away. It's just mean our detection system has stopped working. And therein the problem with indoor mold, that you might walk into a room and say, gee, it smells moldy here. But if that's where you live, and you get used to it, you say, oh, this is a moldy house. You don't smell it all the time. You only smell it when you first perceive it. And it's a phenomenon that I don't know. Excuse me. Yes. We don't necessarily understand the neurology of that.
Yeah, I just had this happen to me last week. I was down at a conference in Austin, Texas, ground zero for toxic mold by some measures, Dripping Springs. That's where Melinda Ballard had her house, and so I walked into the hotel, which I specifically selected because of what it was constructed of, and pictures I saw that didn't have carpet wall to wall, and so it seemed to me pretty low risk on the sort of mold spectrum, on the spectrum of hotels as it relates to mold. And I walked in late, probably 10:30 p.m. and I got hit with that musty smell. And I thought, geez, man, of course, the irony here. And I'm here to talk about mold the whole time. And so I called downstairs and they said, we're booked solid. So, so I had to have a little talk with myself about that, because historically it's not, I don't do well in a musty space. I get, bad dreams and restless sleep and or night terrors in some cases. And so I had a little conversation with myself about acceptance and, but it was interesting as I was, getting ready for bed, I stopped smelling it. And the symptoms were mild. I had a little bit of pressure around my temples, so it wasn't nearly as disturbing as it historically has been. But the point is, to your point, the sense of smell is very quickly numb to these things.
Yeah.
And I think that's how people get stuck in these homes.
Yeah. Yeah.
Were you saying a capitulation?
There are those cynics in the world who say you can get used to almost anything, and a bad smell is perhaps one of the easier ones to get used to.
Yeah. So we ask people if there's a musty smell in their home, and some people say, I don't know. Well, when you first walk in, do you notice it? Do people who come by to visit notice it? And it is tricky if you're living in it. The air is, we quickly take things for granted. Not only is our sense of smell numb, but we quickly take the things closest to us for granted. Is there anything closer to us than air? So it's tough. So I want to get into, your work around, the work that led you to coining the term volatoxin. What was it? What experiments did you run that ultimately led you to that conclusion?
Well, I mentioned earlier on in describing my profession that I'm a fungal geneticist. And during my many years on the Tulane faculty, I taught introduction to genetics and I taught genetics courses. So when I decided to study volatiles, I did something that most people perhaps wouldn't have done. I decided that I would use what those of us in the trade call genetic models. And these are organisms that have been well studied, that we know a lot about their genetic history, that they're easy to grow in the lab. And so we brought in Drosophila flies. A lot of people have studied flies in high school or college, and they're these little flies when they are found in the wild, have red eyes, and a huge amount is known about these organisms. They've been studied for over 100 years. And at this point in time, we've sequenced their genomes, and there have been millions of human hours to understand the way in which Drosophila flies transmit over the generations.
Hey there, sorry to jump in here, but really important. At the end of this video, there's a link that gives you access to a bonus package we put together that has all sorts of great resources. Pro level notes, audio downloads, a fast start guide, as well as some additional resources from our speakers. You will not want to miss out on that. You can also find the link in the description below or at gotmoldsummit.com. So go claim yours today. Now back to the interview.
Because they're insects, they start out as eggs, which turn into larvae, which kind of gross, and then they form pupae. Pupae are like what caterpillar cases are, and then they turn into the fly, and all that happens in about 3 weeks. So we started growing fungi next to these flies, and we set up a method where the fungus was here and the fly was there. And we had a filter so that the fungus couldn't touch the fly. All it could get to the fly was the fungal odors, the fungal volatiles. And sure enough, we were able to kill some flies. And it depended on the fungus. Some fungi would put out volatiles that killed flies. Others didn't. Almost all the fungi put out volatiles that slowed down the fly metamorphosis. It took them longer to get from egg to larvae to pupae to adults, and something we didn't study very much. Sometimes they caused birth defects, and so that the flies that came out at the end had funny-looking wings, or they were kind of crooked or smaller in size. The woman who I had hired on my first grant to study this phenomenon, wonderful person trained in India as a medical doctor named R.T. Inamdar, and she had a PhD in neurobiology from the American University, and she'd worked on flies. And I guess it's a bit of luck, but her PhD dissertation had been studying some pesticides that are suspected to cause Parkinson's disease in humans. And there's a really good genetic model in Drosophila flies where we can understand the genes and we can look at the behavior of the fly for Parkinson's disease. And she saw that some of our volatiles were behaving just the way some of these pesticides, specifically paraquat. And paraquat is known a good bit in the toxicology community. It's believed to be something that contributes to Parkinson's disease. And so we were able to do some very pretty molecular biology to show that one of the major fungal volatiles, a compound called 1-octanthreol, that has a common name, mushroom alcohol, because it smells mushroomy, could cause Parkinson's disease in Drosophila flies. Unfortunately, I've written several grant proposals to NIH to try to move this work into a mammalian system, and it's not been funded. People kind of poo-poo thick building syndrome, but I to this day believe that it is a compound that in certain concentrations may, in certain situations, in genetically susceptible people, cause Parkinsonian or other neurotoxic things. Along the way, we've seen many other conditions, and we've done experiments with mutant fungi and with mutant flies and various combinations. And the more I've done these combinations, the more I've come to believe that some fungal volatiles at certain concentrations can cause toxicity in flies. We've moved the work also into a yeast system, into a plant model called Arabidopsis, and then we did one set of experiments on human embryonic stem cells. And here's an example of why I think fungal toxins can be considered problematic in humans, that most toxicologists accept that it's not good for anyone to be exposed to too many fumes of toluene, which is a common laboratory solvent. In our tests on human embryonic stem cells, 1-octanthreol, mushroom alcohol, was 40 times more toxic than toluene.
Wow.
And
wow.
And so that it was very, very convincing to me. We've only done that one set of of experiments. The state of New Jersey stopped funding stem cell research, and so our stem cell center closed down and all. But it's a paper we published over 10 years ago.
That's shocking.
I want to also point out that with the flies, we've learned that the genetics of the fly makes a difference. And if I were going to project, and I know here I'm on hypothesis territory, but if I'm going to assume that humans also are perceiving and responding to biological volatiles, I think that there's an enormous genetic variation in the person who's doing the receiving. And this is why some people can be in moldy environments and feel just fine and others can't. And what bothers me is that we're very quick to understand that not everybody has an allergy. Some people have peanut allergy. Some people have mold allergy. Why can't we accept that some people might be more sensitive to volatile compounds than other compounds? And so when I talk about it, some people say, well, I've been in moldy buildings. Nothing happens to me. Why is that person saying they're sick? Well, hey, we're genetically different. We don't all have heart attacks. We don't all grow 6 feet tall. I mean, we're very varied as a human species. And so I truly think it's a very realistic hypothesis to, when I talk about volatiles, to also say that the human receiver will have different susceptibilities. And some people are going to be way more sensitive than others. And that it's not a uniform effect. And we understand that about a lot of things. Why we can't understand it about mold compounds, it has to do with probably politics and the insurance industry and some things that I, as a scientist, am not confident to talk about.
Well, there's also a lot of, biases, the cognitive biases at play there, and denial and things like that. People generally have a motivation for saying that it's not affecting them, right? Because someone, it's complex. But, in 2018, Brown University did a rather large study, 6,000 participants, and they found a strong correlation between mold and dampness indoors and depression. This really hit home for me because, as you might recall, my mother committed suicide in the home that I grew up in 2 years after I moved out. And then when I moved out of the house, all my symptoms went away. And yet she stayed behind. And so, of course, mental health is complex.
It is complex. And the more I have read about the human side of mold exposure, the more I realize that we have huge amounts to learn, because there are psychological aspects and there are neurological aspects that are related to the psychological aspects. And then there are just the pure responses that people have to toxic substances, where they will feel weird or, have itchy skin or be nauseated or, as you say, have trouble sleeping. And it's very hard to quantify these things in the varied genetic population with the varied exposures. And because the funding agencies that fund science have so many competing demands on their funds, it's been hard to attract the attention. And I'd heard rumors that the National Institutes of Health might actually be considering putting out some requests for proposals on indoor mold. And I was quite excited about this. It was like 2017 or 2018. Well, we know what happened at the end of 2019. COVID hit. And that sucked so much research focus away from other things that it'll probably be, another 5 years for mold issues to get the kind of attention. The silver lining, though, is that people are more interested in indoor air than they were.
Indeed.
And realizing that many buildings are not properly ventilated, or filtration systems need work. And so I'm trying to look at the silver lining. I think there is now a much clearer idea that we should be careful about our indoor spaces in a way we haven't. And that most of us live indoors. I mean, the statistic that thrown around as 90% of the time. It's more like 98 or 99% of the time for most people. And for babies, it can be, close to 100% of the time, especially in nasty climates where people, the child is living indoors. And as I project from what I've learned from my model systems, I think the populations I worry most about are infants and children. I do not think it is healthy for infants and children to grow up in moldy environments. And I think it can affect their development in ways that we do not understand. And I would really like for people who care about indoor air quality to focus more on the pediatric potential of indoor pollutants.
No doubt about it. So there's a few things to circle back on. Number one, first of all, I want to put a pin in this on the infants and children exposure. There's a significant study from Finland that showed that kids exposed to the musty odor have 2 times the risk of developing asthma later in life. And it was the exposure to the musty smell early in life was the second leading predictor of asthma behind maternal smoking. So it's a kind of a big deal. So I want to talk more about that, but also just to sort of put a bow on and wrap up the conversation around emotional dysregulation, depression. I remember from your work with the fruit flies that they had, you were tracking dopamine production as right, and also some other behaviors around reproduction and things like that. Can you share a little bit more detail there? Because that was a big connection for me when I started Brown University, my own personal history, and then I read your work. I said, this all kind of rhymes here, Yeah.
No, we were able to get fruit flies that had a substance called green fluorescent protein hooked up to the gene, and this is a signal molecule that creates a fluorescent color that can be quantified. And this wonderful postdoc, Dr. R.G. Inamdar, was able to show that in the presence of 1-octanthreol, and that's the only compound we studied, we were able to down regulate, or you stop seeing some of the dopaminergic neurons firing. So there were fewer of them. And because Drosophila is so well understood, they knew just little places in the Drosophila brain where these dopaminergic neurons are located, and with the green fluorescent protein hooked up to the genes involved in dopamine handling, you could see the genes not turned on. And it's published. I think it's good, clean science. The next step would be to try to duplicate that in a mammalian system. And as I said, we weren't able to get funding. I remember one of the reviewers in the rejected grant proposal said, she is not a neurobiologist. And what can I say? I'm a fungal geneticist. I'm not a neurobiologist. I've done some work that I wish that the neurobiologists would pick up. And I think someday they will. I'm a big believer in science, and that I think that there is something called, objective evidence. And I think that scientists are going to, at some point in the not too distant future, start paying more attention to volatile phase biology, volatile phase biochemistry. And it's just a matter of time.
And lots of scientists discover things that aren't within their immediate area of expertise. And so that is the nature of science. If we just stayed in our silos, there would be no real innovation, right? I mean, ultimately, these things, many of the best discoveries are completely accidental. And so, that's a rather narrow kind of classical myopic view. I'm sorry to hear that stopped your funding process. But they also, if I recall, the fruit flies also stopped, the Drosophila flies also stopped reproducing too, correct?
Yeah. Well, we didn't study that very much. And so basically, though, if a fly is dead, they're not going to reduce. And so, so we did not do research. I mean, that's an example of, if people want to pick up on the fly model, there are lots of different things that could be studied, like the effect on reproduction. As I told you, some of the times when we grew flies with not just one known chemical volatile, but with a whole fungus over here, we'd get funny looking flies. The technical term is teratogenic, which means causing birth defects, which, could cause these things. And so there is a lot of potential for studying the teratogenic effects, the birth defect causing ability of these volatiles in Drosophila flies. And not everyone appreciates how many genes we have in common with Drosophila flies and how many human genes are just almost identical to the Drosophila fly ones. And so if it happens in flies, very often you can have a good idea that it might be happening in mammals and other systems. And I have faced that hurdle a number of times in talking about work, and physicians will say, but that's just a fly, and we're mammals, we're humans. But hey, there's a lot of shared biology, and that's why a number of people who've worked on Drosophila have won Nobel Prizes over the 100 years and plus that it's been studied, because it's a really fine genetic model.
No, it's fascinating, and it is a bit humbling, right? When I tell people that we're actually relatively closely related to fungi, I get quite a bit of resistance on that too. So we're a couple of steps up.
As somebody who studied fungi all my life, actually my PhD came from a botany department, because I'm so old that when I was a student, fungi were still considered plants. And it's been about 50 years, but about 50 years ago, when they started studying DNA at a greater level, they discovered that fungi were more closely related to animals than to plants. And for hundreds of years, biologists had just gotten it wrong. And,
amazing.
So fungi are now in their own kingdom. And one of the things I like to tell people is that they're a whole kingdom unto themselves, equal footing to plants and equal footing to animals. However, they don't get a kingdom's worth of attention.
Indeed.
They are an understudied life form.
Yeah, they're tough to study, right? They're elusive. They hide well, and they weave themselves into our world, with the plant and animal world, in such an elegant and almost invisible way. I recently read that all but about 8% of plants have a direct fungal partner woven within them, right? This is fascinating.
Yeah. Well, we hear a lot nowadays about the human microbiome, the microbes that live on us. Well, plants have a microbiome too, and there's been a different vocabulary developed to describe it, and I think that slows down thinking about it. So we now, I think, or many people recognize that we're covered with bacteria. Our insides have bacteria. Also, quite a few fungi in there. Yeasts are fungi. So we have a lot of fungi growing on us. Well, plants have the ratio has changed. They have bacteria, but they have more fungi. Their microbiome is more fungal than the animal microbiome. And so almost every plant is living in close association with one or many fungi. I'd say the many is what's happening.
So we have the fungal kingdom is massive, and most species are yet to be discovered, probably.
Right.
When I get back the dust tests, we're working on a new dust test using next generation sequencing. It's amazing how many species show up as unknown.
Yes.
And so that is really incredible. I mean, it's a huge percentage, actually. And so when we look at mold, kingdom fungi, just for our listeners, mushrooms, macrofungi, microfungi, meaning molds and yeast. So when we're talking about molds specifically as a subsect, approximately how many species are known at this point, and do you have any idea about how many are found indoors or like to grow alongside of us in our habitats?
Yeah, here I'm going to be out of my knowledge zone. I can tell you this, that the traditional ways we've tried to answer that question by growing them on laboratory media has a huge bias in it, because there are a lot of them that don't grow very well on laboratory media or grow very slowly. And so the traditional literature, will maybe point to 40 or 50 regularly isolated species. almost certainly there are way more. And you're seeing this when you do the DNA analysis and you can indirectly measure them. I recently did a big survey of molds from libraries in the published literature.
Hey there, sorry to jump in here, but really important. At the end of this video, there's a link that gives you access to a bonus package we put together that has all sorts of great resources. Pro level notes, audio downloads, a fast start guide, as well as some additional resources from our speakers. You will not want to miss out on that. You can also find the link in the description below or at gotmoldsummit.com. So go claim yours today. Now back to the interview.
And, overwhelmingly, the traditional method of growing molds from libraries, we get certain species over and over again. Actually, one of the most common was Penicillium chrysogenum, which happens to be the fungus that makes penicillin. And famously, it was discovered when it grew in on a petri plate for some microbiologist back in the 1920s. And it's because it's very common in the air and it grows very successfully on media. How many are actually there and grow on things that we don't detect in the laboratory? That's still an unknown, and I'd hesitate to estimate because I feel like I'd underestimate.
Fair enough. So let's go back to mycotoxins, because I think when the tagline of this conference is clearing the air on mold and mycotoxins, and when you talk to doctors or providers that are helping people with mold-related illness or suspected mold-related illness, the hyper focus is mycotoxins,
Yes.
to the exclusion of any other aspect of mold, including the spores themselves. It used to be that it was only spores, right? Allergists only cared about spores. And then somehow we got to this place where I think ever since the big lawsuits in the 90s with Melinda Ballard and the black toxic mold, the whole mold is gold thing, suddenly trichothecenes and their chemical brethren got all of the limelight. I think mycotoxins is a scary word. And you do a couple of quick searches and you go, oh my gosh, T2, this is used in biological warfare, and it's really easy to also forget the fact that antibiotics in many cases are chemically mycotoxins, right? I mean, isn't penicillin technically a mycotoxin?
Well, it all has to do with what it's killing, and I've even written some published papers on that. If it kills something that we don't like a bacterium, we call it an antibiotic, an anti-life compound. If it kills us or one of our favorite animals, then we call it a toxin. It's really a matter of what the target is, what's getting done in, and it's true that in the early days of antibiotics research, when they were screening for compounds that would kill bacteria and kill viruses, and they'd screen in the laboratory, in the early days, they sometimes forgot to, when they found a substance, check if it also didn't kill us. And so there are some compounds that were isolated in the 1940s that were labeled antibiotics that by the 1960s had been relabeled mycotoxins, fungal toxins, because they kill bacteria, they kill viruses, they also kill us, and they kill everything. And so it's a matter of nomenclature and vocabulary, and the important thing is that these are small molecules that are very biologically potent, and they're capable of killing something. And it's the selective aspect. There are some compounds that are selective, and they'll kill a bacterium, and they won't kill us. And those are the ones we think are the good drugs. If they kill both the bacterium and us, then we call it a toxin.
Yeah, those guys are bad guys, and they get demonized, of course. Listen, we should do our best to avoid these compounds. And so you brought up aflatoxin, and I don't get a chance to talk to aflatoxin experts every day, and so you brought up the fact that first of all, aflatoxin made by Aspergillus flavus, right? Hence afla, right? Right. And so commonly grown. We see a lot of aflatoxin in peanuts.
Yes.
Right? Peanut farmers have a pretty high rate of certain kinds of cancers. Is it pancreatic cancer? I believe.
I don't know. I don't know.
So the question I have is around this. Is aflatoxin, does aflatoxin become airborne? Do mycotoxins in the actual, not to be confused with volatoxins, the volatile organic compounds. I'm just saying that for our audience. Do mycotoxins become airborne easily?
Well, they're usually found inside the cells, the cells of the fungus, which are called hyphae or spores, and it's hard to get a huge amount of mycotoxin up in the air and the aerosolized spores and hyphae. It's not impossible, but it's difficult, and that's why back in the 90s I used to testify for insurance companies and say, hey, I can see this person's lived in a moldy house and they're sick, but I don't think it could be caused by a mycotoxin. And for example, aflatoxin, it doesn't, the fungi that make aflatoxin don't grow well on building materials. They grow wonderfully on corn. They grow wonderfully on, in fact, a huge variety of our foodstuffs. And that's why nowadays when you do an aflatoxin test on human urine, it's quite common to find trace levels of aflatoxin and certain other mycotoxins. I believe that the vast majority of the time, those mycotoxins are coming through our diet, that we're eating low quantities of them in our foodstuffs because these molds are competing with us for the food. The mold likes the same stuff that we do. I think it's possible, but very unlikely to be exposed to mycotoxins through the air and through absorption in our skin. It could happen, and I think there are environments where workers are exposed to huge levels of respiratory and skin contact with molds. But in most indoor air situations, I don't think that mycotoxins are a problem. And so there are experts out there who testify and say the same thing I'm saying. Where I'm different from some of those experts is that I don't give the fungi a pass. I say, fungi don't just make toxins. They have these cell walls. They have fragments that they have. There are many aspects to fungi other than the mycotoxins that could potentially make people sick, and that we shouldn't throw away the whole fear of indoor mold just because the toxins are unlikely to be the culprit.
So this is, first of all, I have to ask you this. This is, it seems like something that you've thought quite a bit about. Do you regret the defense work that you did back then?
I do, in the sense that I was doing a disfavor to the plaintiffs. I don't in the sense that I learned so much about indoor air. And I would not be doing the research I'm doing now if I hadn't learned about indoor air problems. And I had been studying agricultural products. I collaborated with the Department of Agriculture for over 30 years. And so my focus had been very much on the food supply. And rightly so. I mean, toxins in the food supply to this day, they'll be with us forever. We don't want to eat toxin-laden foods. I would not have known about the indoor air problems if I hadn't taken those cases. And it wasn't a huge number of cases, but it was some. I let down some people who I never met, but I learned something that I hope is going to help others in the future. And I wouldn't be doing the research I'm doing now if I hadn't had that legal exposure. I also sent a son to an expensive private college on the money I earned. That's a purely unrelated thing.
Well, I think you're helping a lot more people with the work that you've done since. And so this is just the sort of the nature and evolution of learnings.
Yes.
But, I have also struggled with these mycotoxin urine panels. I see a lot of people come to us with these high numbers,
right.
And, aflatoxin or Crotoxin A, DON. I always mispronounce it. Deoxy.
Valinol.
Well, no, yeah, thank you. And there are a number of them that just a quick search, you quickly see these are very, very common in food.
Yeah.
And people want us to find the mold in their house that's making this toxin, as if we're going to go in and target this one species and eradicate it, which is, kind of entertaining, but also typifies the confusion around this, as if mycotoxins is the only source of human illness. In my experience, after doing this 20-something years, and using dogs and guiding people through remediation and doing lots of testing, both for VOCs, mVOCs, spores, and everything in between, is that there's the distinction between building-related exposure and potentially food-based exposure. And so building-based exposure to spores, potentially small amounts of mycotoxins, and the volatile organic compounds, the musty smell, being the source of potential discomfort, disease. And then the food-based exposures in terms of mycotoxins. And I have this Venn diagram in my mind's eye where there's an overlap there. And of course, and where that could potentially be the overload that really amplifies symptoms for people. And so it's not to say, because I tend to push the mycotoxins to the side in the conversation, because I don't think it's, people call us about their buildings. I'm not here to help them with what's in their pantry, but I do encourage them to really look at that, because it's imported nuts, seeds, grains, spices, conventional meats and dairy even, right? Because of the animals eating the moldy grains. The distinction that I want to make is building related exposure versus food-based exposure, and bring some clarity around that, so that people don't, when they get one of these mycotoxin urine panels, first of all, if they're excreting it, good news. These things don't excrete well, right? And so if you're not excreting them in modern America, you may have a problem, because your chances are you are eating it, and you may not have the detox pathways working. So there's a lot of sort of counterintuitive stuff with this, right?
Yeah. Well, I've talked to some of the people who do the urine panels, and here's the case where we need a government study. We should have somebody out there who samples a lot of, quote, control people who don't have any mold problem in their house versus people who claim to have a mold problem, and see if both populations have similar mycotoxin panels in their urine. And no one has funded that study, and it would be quite expensive to do, and to recruit all the people into the study and to do all the testing and blah, blah, blah. And it just hasn't risen to the level for funding agencies to think it should be done. But what you're saying in terms of just using our brains to think about it, and I had never necessarily thought about the Venn diagram way of going at it. But if you are, as many of us I believe are, consuming low levels of fungal toxins in your diet, and you're also being exposed to molds and their metabolites and their structural components in your home, it could have a synergistic effect and make things worse. But they're not necessarily the same thing. And we scientists, people make fun of us for saying we need more research, But there are areas which are less studied than others. And there's a great deal we do not know about indoor air quality and indoor air exposures to not just fungi, but to all sorts of things in our environments. And a toxicologist could be more articulate than I am, but we use now chemical building materials that weren't used in the past. And I distrust many of those contemporary building materials. They may be water repellent, but all those fluoridated compounds that go into water repellency, there's a lot of evidence that they're not good for us. So we're being hammered as contemporary people by traditional toxins, such as mycotoxins made by living things, simultaneously with chemical toxicology due to the power of modern chemistry to create plastics and other convenience elements. And it's when you start thinking about it, it's very hard to disentangle why an individual might be having a carcinogenic response or an allergenic response or a psychological response. And it's but we don't live in as clean a world as we like to think we do.
No, and to do these studies, you know, I mean, how would you really control for
Yes.
the exposures of modern day? You know,
Yeah.
I people come to us all the time and say, hey, you know, I've got these symptoms. I've been sick for a while, and we think it's the building, but if you track back where their symptoms began, oftentimes a big chemical exposure they become suddenly sensitive to mold, or a big mold exposure and they suddenly become sensitive to fragrances and things. So there's this cross sensitization that is fascinating. And I the way I see it, of course, I'm not a scientist, but I've been doing this for a while and seen a few thousand cases, is that the body doesn't know the difference between the microbial volatile and the man-made volatile. And it just goes, you know,
I agree.
too much.
There's something going on, and there are huge numbers of people with these so-called multiple chemical sensitivities, and it's real. And I get very angry when I meet physicians who just say, "Oh, it's all psychological," and often they do it to women. It's all in her head. Well, it's not all in her head. You know, that there's so many people who are truly suffering from these chemical sensitivities, and it infuriates me that too frequently the medical profession just rejects it as some kind of psychological quirk. It's a real organic thing. We don't understand it, but it's real.
Well, it may be in your head in the sense that, and the more research I do on this, the more research I do on the research being done about the trigeminal nerve, which is not a conversation that we should really unpack here, but the nerves in the face and their ability to sense chemicals at very, very low levels in conjunction with the olfactory sense is an area of great interest for me.
Yeah.
And so those nerves do live in your head,
Yes,
but it may not be in your mind, so to speak.
Yes. And, we don't want to go down the whole pathway of psychological diseases and the difficulty of classifying them. It's difficult enough to classify these mold-related diseases without worrying about it.
So, before we pressed record, we talked a little bit about a pediatrician that you met when you were at a recent IAQ conference in Germany, and we talked about a little bit about infants and toddlers, infants and children and the exposure. Are you seeing some interesting insights? Are you seeing more interest in that area? What was that conversation like? What did that pediatrician have to say?
There was concern, but I can't say that it had mounted to any great interest. Apparently, the pediatrician had recently read a study from Poland in which people had measured IQ of children who had grown up in substandard housing versus good housing, and that there was a significant difference in the IQ of, I didn't read the paper, so don't quote me on this, but let's say they were 10 years old, and that the children who'd been in this substandard housing, and that there'd been an attempt to match them on other things other than the mold. It's very tricky research, but just use common sense. I mean, people do not want to raise their children in toxic environments, and substandard housing where you can see mold growing on the walls or surrounding. Most of us have the realization that this is not a good place to raise kids. And small kids, they crawl around on the floor, they put things in their mouth. So it's not just a matter of volatiles. It's a matter of a growing organism putting down nerve cells, putting down body parts, growing all the things that are going to be important when you become an adult who has to function in the world. And if exposed to things that are not healthy, it doesn't take a whole lot of research studies to recognize that we don't want to do that to children. And I think that the pediatric community would agree. Pediatricians worry about children all the time. Many of us who are do it while we're parents of young children, and then we sort of forget about the youthful population. But
hey there, sorry to jump in here, but really important. At the end of this video, there's a link that gives you access to a bonus package we put together that has all sorts of great resources. Pro level notes, audio downloads, a fast start guide, as well as some additional resources from our speakers. You will not want to miss out on that. You can also find the link in the description below or at gotmoldsummit.com. So go claim yours today. Now back to the interview.
Pediatricians are some of the nicest people I know, and I think this focus on the welfare of children is something that we should always keep in mind when we're talking about indoor air quality. That if we're not doing it for ourselves, we should do it for our children. We don't want to mess them up developmentally.
Yeah, no doubt. So we're talking about mold, but of course, and we talked about chemicals, man-made chemicals, and you really can't have a conversation about healthy buildings without speaking about both of them almost in the same breath. And I guess just it's tangential, but so closely related that I feel like I want to just drop this in here, which is that we build our buildings out of mold food. So they get moldy very quickly when they get wet. And then we also slather them with petrochemicals in the form of paints and finishes. And so when we're talking about children crawling around on the ground, yes, they're going to be exposed to spores. And chances are, actually, that may not be such a bad thing because, of course, these can be hormetic, right? And they can train our immune system. But the other stuff that lives in dust is pretty nasty. So when you start looking at buildings shedding particles, why is it that you have to refinish your polyurethane floor? Where did the polyurethane go? Why is it that you may have to repaint that wall? Where did the paint go? And it goes into these tiny little bits that end up in the dust in your home. And so your children and your pets in particular crawl around on the floor, and they ingest it through something called incidental ingestion, up to 100 milligrams a day by some measures. And these are very potent, very dense chemical particles that are not to be consumed, not to be breathed, not to be eaten, and children and pets get a disproportionate concentration of these per pound of body weight. And so it's a big deal, right? So it's not just to keep your house clean, and not to just keep it dry, but keep it clean. And so it's not just enough to run HEPA filters, but to vacuum these corners up, right? Clean this stuff up so that the dust, not the biological dust, the non-biological dust, doesn't become part of their biology. So anyway, that's a little side commercial here for watching out for those.
With you. All of us who get interested in toxicology have to face the fact that we don't live in, unipolar worlds, that we are, faced with just huge plenitude of chemicals and potential toxicants at one side. Those of us who are scientists recognize that we have to be reductionists. We can't study all of them. We'll go crazy if we worry about all the variables. And I've chosen to focus in on this subset of chemicals that are volatile phase molecules made by fungi. I recognize that they are just one of the many potential toxicants out there that can cause a problem. Because I love fungi and because I am fascinated by the chemistry of fungi, I will stay in my little bailiwick and hope to add some small amount of knowledge to the information pile. But there's a lot of other stuff out there, and I recognize that the burden of synthetic chemicals may be larger for most people than the mold volatiles. And I back up what you're saying, that cleanliness is a virtue that you can make fun of. We like, I'm a slob and everything, but if you're raising children, I think it's important to make sure that you don't expose them to too much bad stuff. And there's a lot of bad stuff out there in the modern world, a lot of bad stuff. And chemical industry has brought us miracles, but it's also brought us a lot of carcinogens.
Yeah, no doubt. And also, just to clarify, not such a bad thing for kids to be exposed to mold spores in a normal quantity, a background, the kind that the amount you'd find outside. It's the mold growing indoors that's the problem, not the presence of mold spores in dust or in the air. That's, I think, a really important point. I'm much more concerned about the man-made stuff in the dust than I am about the biological stuff.
Yes.
In the dust. You know.
And what it is when there's massive mold growth, and you know, we haven't talked that much about the quantity, but when you see a home that's been mold damaged through leaks or whatever, you recognize that it's a different order than some things. Or when you needed the mold sniffing dog, sometimes the mold growth is hidden, and it can be in insulation and just, you might smell the musty smell. It's only when you break the wall open that you discover just how much mold is growing. As you say, we build our houses out of mold food. And I love the whole organic movement, but I worry about people who decide that they're going to insulate their home, say, with cotton, because, you know, if it gets wet, you've got a potential disaster. So better idea to do that if you live in the desert in Arizona than if you live in Houston, Texas, where you have hurricanes all the time.
Yeah, no doubt. You know, I'll tell you a kind of an interesting story that this has just happened a few weeks ago, where one of my potential partners, a commercial partner, collaborative partner, who's pregnant, was concerned about mold in her house. And her husband lost his job as a contractor, union contractor, and he decided he was going to take his extra energy and do some renovations in the house. And she worked from home, and she called me up because she was very concerned about having some extraordinary symptoms after her husband opened up the wall, and specifically, she started having all these nosebleeds. And so I said, well, you've got to, first of all, go somewhere else immediately, and also send me pictures of what's going on. And so the pictures were, you know, at least 100 square foot of what I was, you know, I've seen enough of it, Stachybotrys, Cotomium, you know, the usual suspects, right? Now, without taking any samples, I don't want to leap to, you know, speciating, but it was very clearly the same kind of suite of typical chronic water damage molds. And so I instructed them on what to do next and how to remediate it, because they didn't want to bring in professionals, and he was a contractor. So I gave him some guidance around the best practices according to the industry standard. And so they went about doing their best to mitigate and remediate. And she was obviously very concerned about the nosebleeds, as was I, since trichothecene is hemorrhagic, right? The toxin that Stachybotrys produces will cause blood vessels to potentially burst. And so there's many anecdotes about this. And I in fact once went into a very moldy space, floor to ceiling. I mean, literally 1,000 square feet of black mold. I was there to take samples to document this problem for to validate the house as being uninhabitable, so that ultimately they would be able to just, you know, knock it down. And so I went in there in my youthful arrogance, without any respiratory protection, and I reached down and I sneezed, and then I had a big inhalation, and my next breath out, my nose burst, and blood went all over the place. I don't get nosebleeds. I haven't had nosebleeds since I was a nose picker when I was 5 years old. You know, like it just doesn't happen to me. And I went outside and I stopped the bleeding, and I was humbled by that, because I realized instantly what had happened based upon the literature I'd read. And so I want to highlight something here for everyone who's listening, because here Joan and I are talking about mycotoxins not being airborne, and also that the food is probably a primary exposure and all of that stuff. These are two circumstances where I have seen people develop acute symptoms that are directly potentially related to the mycotoxin itself. But the distinction here is huge amounts airborne, right? Huge amounts, direct exposure, not the kind of mold that might be growing in a wall where there's no direct exposure to the spores or the toxins. If you don't have a huge visible amount of mold and it's been disturbed, for example, in the middle of a remediation project, or in the middle of a renovation where you thought it was just a renovation, and next thing you know, you find Pandora's box, right? These kinds of exposures can be very, very problematic and can lead to really serious illness. And I want to make that distinction. Is that fair? Because
I'm glad you said that, because we don't want to deny the fact that trichothecenes can cause direct skin contact with very, very negative effects, and so it happens. We're talking about the cases that don't have that, and the fact that people sometimes use the fear of the trichothecenes to rip off homeowners, do unnecessarily remediation, and then the fear that some people have that is unfounded because they have had some ochratoxin found in their urine sample, and it probably came in through apple juice or something they drank, not through breathing mold spores. And so we're talking about lots of different things at the same time, and we've also thrown in the synthetic chemicals that can cause problems. But I think that the take-home lesson is that I happen to believe that most people are pretty smart, and if given the facts, can recognize truth from fiction, or truth from bias, or truth from somebody who's just trying to sell you a product. And that I feel that we should make mold education part of the education of studying indoor air environments. It's not the only thing that's important in indoor pollution, but it's one of the things that's important. And when mold grows excessively, as I think it will be more and more with global warming and hurricanes and unexpected floods, that people should be aware that it can be a potential danger. So,
no doubt about it. I often think that mold and how to deal with it should be taught much earlier, since we live on, planet fungi. We live on a water planet. We all live in buildings and breathe air. There are a lot of things that are a lot less practical that we're taught in school. This affects nearly all of us at some point in our lives. So I guess trying to land this plane, because it's always such a pleasure to talk with you. We can talk for hours, as we sometimes have. I'd like to know, actually, on a personal level, what is it that you do? Do you have any sort of a mold avoidance strategy? Do you watch the foods you eat for mycotoxins? Is this something that's on your radar on a personal level, just out of curiosity?
Well, no, I do try to make sure that my home doesn't have any leaks, and that I don't have a mold problem in the spaces where I live. When it comes to food, I happen to like to cook, so where I'm careful about my own food is probably not so much on mold, but not so much on synthetic preservatives. And I like to eat a lot of fresh fruits and vegetables, and I avoid processed foods, but that's not a mold issue. And if I see mold growing on my food, I just throw it away.
Well, it seems that avoiding processed foods is a great way to avoid a lot of mycotoxins in food, since this seems to be where a lot of it comes from the imported grains, and that are very inexpensive, and that find their way into all the ultra highly processed stuff, all the boxes and bags are in the middle of the grocery store. So fresh seems like a culinary approach to things, where you're eating fresh food, organic food, seasonal, grass fed, and pastured meats and things like that is a pretty good strategy.
Right. I
Yeah.
agree.
Okay.
I've always felt lucky. I'm a woman who's raised a family while also working full time, and I've always felt lucky that I like to cook. That I have come home from a full day of work and cooked dinner for most of my life, and I still enjoy cooking. I find it therapeutic. But again, that's a whole other story. And I'll tell you this: that I cook quite a few macro fungi, and I think they're good for the diet. And I eat more mushrooms than most people do.
Yeah, no, and I think that's also important. A lot of people throw the baby out with the bathwater, and when they're trying to avoid mold, especially if they've had mold-related illness, they're often told to avoid mushrooms, which I think is silly, since mushrooms are so well known to do wonders for the immune system.
Yeah.
Yeah. So, all right. Well, I guess in closing here, how do people learn more about your research?
Oh, I guess listening to things like this. I have never been one much to seek publicity outside of the scientific community, and I admit that I'm somewhat uncomfortable when I do things like this. I'm much more comfortable giving a lecture to a university audience than addressing the public. I've done a lot of addressing my fellow scientists, and that's my niche in this world. Jason, it's your job to bring the news out to the general public. It's my job to continue doing the research on my genetic models and try to make more biologists aware that there's activity in the gas phase and that we should be paying more attention to the things we smell, both good and bad.
Well, so I will direct anyone who's interested in learning more to Google silver linings and Joan Bennett for a beautiful paper written in memoir form, something I don't think you'll probably ever see again or anytime soon. It's a fascinating read, especially as scientific papers go. So I guess with that, what I will say is, Joan, we've been friends for a long time, and I'm grateful for our friendship. Your work has truly opened my eyes to help me really to get a much greater understanding of what's known out there, and help me to put the pieces of the puzzle together. And it's helped me to help thousands of people. So know that you've directly impacted those people via me, and that. Yeah, it's very real, and through this, I think you'll also help dial a couple of people down, talk them from the ceiling, right? Get them to stop and pause and think. These mycotoxin urine panels, maybe it's not. Maybe we need to pump the brakes. Maybe the data needs to be interpreted or looked at a little bit differently before we start doing things like tearing our house apart or inviting people in that have huge financial motives. And so just slow the roll a little bit, But thank you very much. Thank you very much for your generous contribution. I know this is not a comfortable thing for you, and so I'm even more appreciative of your willingness to participate in our summit. It means the world to me.
Thank you, Jason, and good luck. Keep up the good work.
Thanks, Joan. Until next time.
Okay.
Hey, before you go, in case you haven't done it already, go to gotmoldsummit.com to claim your bonus package today. It's filled with all sorts of great stuff. Audio downloads, pro-level notes, fast start guide, and some additional gifts from our speakers, all absolutely free, so you won't want to miss out on that. Again, go to gotmoldsummit.com to claim your bonus package. You can also find the link in the description below.